Good morning, everybody. I'll be talking about analysis of the spatial specificity of bold and cerebral blood volume fMRI in order to get reliable, optimized detection, decoding, and high-resolution imaging of cortical columns. Outline, I will give two examples of cortical columns, orientation and ocular dominance, and how we measure them with neurophysiology and optical imaging, a model for simulating fMRI of cortical columns. I'll speak about spatial specificity of the gradient echo and spin echo bold responses relative to neuronal activity with data from human area V1, spatial specificity of bold and cerebral blood volume responses in gray matter and blood vessels with data from cat area 18, optimization of high-resolution fMRI, and likely there will not be time to talk about mechanism of decoding and what can we learn from that on mechanism of high-resolution fMRI. In the second part of this talk, my former student, Dennis Chaimov, will present here a toolbox in order that helps to plan experiments for optimizing experiments of high-resolution imaging of cortical columns. So I'll start with examples of cortical columns in neurophysiology and optical imaging. Neurons in the visual cortex in area V1, but not only in area V1, are sensitive. They show preference to specific orientation of bars. Hubel and Wiesel, awardees of Nobel Prize, used experiments or performed experiments in cats and monkeys. They presented bars of light, oriented bars, and moved them back and forth while recording neurophysiology. We can see here the tip of the electrode close to a pyramidal cell and recorded spikes. So in this case, for this cell, when the bar is horizontal, we get almost no response. Obviously, the duration of the stimulus is where the dark background is. When the stimulus is right oblique, we see a higher response. When the stimulus is vertical, we see a very significant higher response. So this cell shows preference to vertical orientation. We can use optical imaging of hemodynamic and metabolic responses used mainly in animal models, but sometimes also in the neurosurgery suites with human patients. We expose the cortex by removing the skull and the dura mater, so that would be an image of the cortex taken directly by a CCD camera, shine light alternately of two wavelengths that help us compute changes in deoxyhemoglobin that represents blood oxygenation, bold fMRI, and changes in total hemoglobin that represents cerebral blood volume fMRI. Now, the physiological response is absolutely identical to the physiological response of fMRI. What is different is the measurement, the principle of measurement. In optical imaging, the principle of measurement is the different absorption of oxy and deoxyhemoglobin as a function of wavelength of illumination. In fMRI, the principle of measurement, if we talk bold fMRI, is the changes in content of deoxyhemoglobin. So we can use this system in order to image this to oriented bars, and we get in each pixel a so-called tuning curve as a function of the orientation. Now we can go pixel by pixel and compute the vectorial summation, and we show in color code that is represented here the preferred orientation for that pixel. Now, why do we call these columns? Because if, for example, we mark 6 locations on top of cortex and insert an electrode and record action potentials from these locations, we can see that the preferred orientation as a function of cortical depth is approximately constant. It's invariant to depth. And it matches what we get with optical imaging from top of cortex. So that's why we call them columns. There are also columns, well, there are several types of columns in visual cortex, somatosensory cortex, auditory cortex, but in visual cortex, another common columnar system which is being imaged is that of ocular dominance. We can see here again results from optical imaging. That would be the image of cortex, and this would be the difference in responses to stimulation with the left eye and right eye in the monkey. The area with the stripes is V1, the area without the differential response to ocular dominance is V2. Humans have ocular dominance columns as well, as can be seen by post-mortem cytochrome oxidase histology. So the input from the left eye and the right eye is segregated in stripes in area V1, and the post-mortem histology here was obtained from a patient who lost one eye while being already adult. And so the only eye which is metabolically active is the right eye, or the input from the right eye causes stripes in V1 that are metabolically active. So whatever is brown here represents input from the right eye only. We can see here, according to the scale bar of 1 millimeter, that these ocular dominance columns are very fine scale. And that's the challenge in imaging cortical columns. They are a fine scale. However, fMRI can image ocular dominance columns, as shown here, with gradient echo and spin echo imaging. All this data is from one single subject in 6 different imaging sessions, 6 different days. So 3 gradient echo, 3 spin echo, and we see the differential response to the left eye and the right eye. The 4th column shows the end operator, the logical end, from 2 days of imaging, and the 3rd column shows the logical end of 3 days. So we see that not only we can image them, they are also, the imaging is reproducible over 6 days and 2 different imaging modalities. So that was about orientation column, ocular dominance column. Now I'll present a model for simulating fMRI of cortical columns. And using the model, I will estimate the spatial specificity of gradient echo and spin echo, bold responses relative to neuronal activity. So here's the model devised by Denis Chaimov. We follow Roger and Schwartz by starting with Gaussian white noise, an image of Gaussian white noise that we filter with anisotropic filter in order to get stripes that are similar in topology and structure to ocular dominance columns in humans. We simulate neuronal responses to stimulation of the left eye and the right eye, and we convolve with the hemodynamic bold point spread function in order to mimic the bold response. Lastly, we simulate K-space sampling to get the voxelized results. Now, we want to use this model. We can feed this model to data and extract specific parameters. In this case, we are interested in extracting the point spread function, not of the imaging process that was presented by David Norris earlier, but the point spread function of gradient echo bold and spin echo bold, the physiological bold point spread function. So point spread function can be defined or is the spatiotemporal fMRI response to a transient point-like event of elevated neurophysiological activity. Measure of spread are usually standard deviation or full width at half max of the Gaussian, and obviously when standard deviation is longer than or larger, then we can see that the Gaussian is wider, represents a lesser spatial specificity. Now, the bold response is cortical position dependent, and this raises the question, do we care about the point spread function? We do care because quantification of the average tangential point spread function is useful for comparing the spatial specificity of functional MRI contrast and for planning, optimizing, and modeling fMRI at high resolution. In addition, we will specifically focus on the point spread function of differential imaging, of taking the difference between responses, which is more homogeneous, parallel to cortex. Previous studies of the point spread function of the bold response were carried out at 1.5, 3 Tesla, and 7 Tesla millimeter, and you can see that the point spread function at 7 Tesla reduces the point spread function, meaning the spatial specificity increases. The most recent study by myself estimated less than 2 millimeter full width at half max using gradient echo and single condition results. So here is how it looks. All previous point spread function studies use retinotopic stimuli, and using retinotopic stimuli, they had information on the falling edge, the spatial falling edge of the response, which can be used in order to fit a Gaussian or a sigmoid function, and this way get a Gaussian and estimate the full width at half max. However, all previous studies on the bold point spread function assumed a point-like neuronal response. Neuronal responses are never point-like. As we can see here, the scatter of receptive fields measured by Hubel and Wiesel, we see a significant cluster in V1 when inserting an electrode orthogonally to cortex. The scatter is approximately 2 millimeter, so we need to take into account the scatter of the neurophysiological response if we want to look only at the bold spatial specificity. So we want to estimate the point spread function of differential hand spin echo bold and gradient echo bold response in humans at 7 Tesla while avoiding the neurophysiological spread confound. Now, in order to avoid the neurophysiological confound or spread confound, instead of using retinotopic stimuli, we used the data of ocular dominance columns, differential ocular dominance responses, and took into account information that we have about the neurophysiological response in V1 from monkeys in order to determine the point spread function. So we quantify the point spread functions that best predict the spatial structure and magnitude of differential ocular dominance responses. So we took the model and fit it to ocular dominance obtained with gradient echo and spin echo. Because it's quite a high-dimensional model, it is very useful to constrain it with a priori data on the distribution of features of the ocular dominance columns. To do that, we obtained, or we estimated, parameters of the ocular dominance columns from post-mortem histology. We can see here distribution of spatial frequency of the ocular dominance columns, the irregularity. So this would be in green, a very regular pattern, and in yellow, a very irregular pattern, and the branchiness of the ocular dominance columns. And that's how the fitting looks. In the main box here, we have the model. The model generates, iteration by iteration, a simulated ocular dominance map that we can convolve with the hemodynamic response function in order to obtain differential ocular dominance columns on the left that we compare to the differential ocular dominance columns from a subject on the right. And in each iteration, we ask, what is the probability of the current parameters given the data? And as part of that, what is the probability of the current estimated point spread function width as a function of data? So that's how it looks, the fitting of the model to the data. On top here, we see two differential ocular dominance responses with green echo and spin echo from the same subject. On the left is a noise pattern, iteration by iteration. Then the common ocular dominance pattern obtained for that iteration. And here we'll see the model gradient echo and spin echo differential map that we want to converge to the data. If it converges to the data, we are in the right direction. And on the right, we will see the point spread function obtained with spin echo fMRI as a function of gradient echo fMRI. We start with 2 millimeters. So let's see how it goes. The first iterations. And then pretty quickly, the model converges on patterns that are very similar to the data. And the point spread function, the probability density function that we estimate for the point spread functions, goes to approximately 0.8 for spin echo and approximately 1 full width at half max for gradient echo. Can you advance one slide for me? Seems that I cannot do it from here. Technical help, please. Technical help. Can you advance one slide? Maybe with the keyboard. Can we advance one slide, please? Okay, so these are the probability density functions that we get, and we estimate for gradient echo point spread function the full width at half max is 0.99 millimeter for spin echo, not much narrower, 0.86. We can move on now and speak about the spatial specificity of bold and cerebral blood volume responses in gray matter and blood vessels. I'll emphasize more the gray matter because of time limitations. Data obtained from cat area 18. So with optical imaging, we can see here the cat area 18 cortex. The dark curves are PL blood vessels, image with 530 nanometer and 617 nanometer. We obtain this way changes in deoxyhemoglobin content and in total hemoglobin that represent bold and cerebral blood volume functional MRI. We can obtain a mask of the blood vessels, in this case by an algorithm of nearest neighbor learning, and split them to arteries and veins. We can also estimate the width, the diameter of each of the vessels in order to learn how the changes will refer to the diameter or the spatial specificity, how it refers to the parameter, to the diameter of the veins and arteries. And here is a slide that already, just by looking at the results of the map, shows the difference between the spatial specificity of deoxyhemoglobin or bold in the top row and total hemoglobin or cerebral blood volume at the bottom row. So we used gratings, moving gratings, differential imaging, meaning the difference between the peak response and the response to the grating, which is 90 degrees relative to it. And you can see that with bold imaging, we get fuzzy, unclear differential responses. In contrast, with blood volume imaging, we see fine scale imaging of cortical columns at a relatively high amplitude. We can then look at the contrast to noise ratio in vessels and see that with small vessels, the contrast to noise ratio of veins is very similar to the contrast to noise ratio of the gray matter when looking at bold response, and this is true also for SVM support vector machine decoding rate. This is not the case for blood volume imaging, where the response in blood vessels drops, the spatial specificity drops relative to gray matter, both as can be imaged by contrast to noise ratio and support vector machine decoding. Interestingly, with cerebral blood volume, we get selective responses also from arteries, which we cannot get with bold imaging. And overall, if we look at the spatial specificity of deoxyhemoglobin or bold divided by the spatial specificity of cerebral blood volume, we see that in gray matter, the ratio is 0.55, meaning the spatial specificity of cerebral blood volume is much better than that of bold imaging. So I'll get to the main conclusions here of this study. For high-resolution cerebral blood volume fMRI, the smaller the diameter of contributor vessels, the higher the spatial specificity. The spatial specificity of cerebral blood volume with no use of contrast agent. So I'm saying with no use of contrast agent because previous studies showed similar phenomenon while using a ferrum-based contrast agent, Myon. Here it is based on just the intrinsic blood responses. The spatial specificity of cerebral blood volume is superior to that of bold imaging. So we move on for demonstrating a model for optimizing a fMRI of cortical columns. A toolbox is available on GitHub. That's the toolbox that Dennis will soon present here. We took again the model and we varied spatial scale and irregularity of the columnar pattern. So you can see the spatial scale, the spatial frequency on the vertical axis. Irregularity changes on the horizontal axis. If you look at the second row here, the pattern to the left is very irregular. The pattern to the right is very irregular. We varied the point spread function of fMRI response, the voxel size, and the signal-to-noise ratio, considering both thermal and physiological noise. And here are some findings. All measures associated with multivariate detection and decoding could be approximately calculated from a measure we termed multivariate contrast-to-noise ratio, which is a function of the contrast-to-noise ratio and the number of voxels. So the contrast-to-noise ratio and the number of voxels is a very important measure in obtaining or predicting the results of decoding and detection of cortical columns. The multivariate contrast-to-noise ratio implies that optimal voxel width for detection and decoding is independent of changes in response amplitude, SNR, and image volume that are not caused by changes in voxel size. Also, what matters for a success in imaging cortical columns, decoding of cortical columns? It seems that the bold response point spread, the point spread function of the bold response is the determinant factor. You can see that at 3 Tesla, we get flat, absolutely no success in decoding real cortical columns or imaging real cortical columns, mainly because of the point spread function being wide. And this is not the case for the amplitude or the field strength dependent noise. So the bold point spread function is the determining factor in successful decoding or imaging of cortical columns. The voxel size, the optimal voxel size depends on the aim, i.e. detection, decoding, or high-resolution reconstruction of the columnar pattern. We can see that the voxel size for univariate detection of cortical columns is approximately 1 millimeter for spatial frequency of 0.5 cycles per millimeter. For the same spatial frequency, the multivariate, the optimal voxel width for multivariate decoding is also 1 millimeter. But for reconstructing, for really describing in detail the pattern of the map, we need a smaller voxel size, in this case, approximately a 0.6 millimeter. Lastly, for regular patterns, optimal voxel width for detection, decoding, and imaging, the pattern of cortical columns are close to half the main cycle to the Nyquist sampling voxel size of the organization. For irregular patterns, optimal voxel widths were less dependent on the main cycle length and differed between univariate detection, multivariate detection, and decoding and reconstruction. So lastly, how do we know that, suppose we work on obtaining a map in an unknown area, a cortical map of columns, how do we know that we use the adequate voxel size? So here is a simulation of a cortical map, and it's very common to have a Gaussian-like frequencies of the organization. Now, an adequate voxel width will still give us, in the spectrum, a contrast-to-noise ratio, which is approximately a Gaussian, with no interruption. If our voxel is too large, we will get a cropped Gaussian. It will not go back to the baseline of the responses. It will be cropped, either here, for example, or here. So that's a good way to know whether we are using a good voxel or adequate voxel size. So I would like to thank my students, Denis Chaimov and Zeshan Yao, my collaborators, Isa Yakub, Kamil Ugobil, Sengi Kim, Mitsushiro Fukuda, and Changhong Mu. And I would like to thank you for your attention, and I would like to invite Denis Chaimov to demonstrate the toolbox for optimizing fMRI of cortical columns.